Hi, assalamu alaikum everyone. My name is Naseel, and today I'll be talking to you about acid-base balance, so what normally happens, and then the disorders. Alright, so this is basically three lectures, your physiology lectures 9, 10, and 11 combined into one session. So for what we'll talk about today, we're going to talk about an overview of the normal acid-base balance, and then we'll focus on the bicarbonate and carbon dioxide buffer. Then we'll talk a bit about normal metabolism. Then we'll go on to the main interesting topic of acid-base disturbances. And finally, we'll touch upon the anion gap and we'll do some practice questions. Okay, now because this session has a lot of thinking, it's going to be pretty interactive. So anything you want to answer, you can either unmute yourselves or use the chat. Okay, I have it open on my phone. And if you have any questions at any time, you can also use the chat or unmute yourselves and talk to me. Okay, so let's get started. Now, what is pH? Okay, now we've heard this term a lot in chemistry, high school chemistry, and now we're hearing it in renal, right? So what is it? Okay, it is basically a scale. Okay, it's a scale that goes from 0 to 14, and it measures how acidic or basic a substance is. Another word for basic is alkaline. Okay, now your neutral number is at 7. Okay, so a pH of 7 is neutral. Water is neutral, so water has a pH of 7. Less than 7, so going all the way over here, this is acidic. This means that you have more acidic things like hydrogen ions. Now, above 7 is what we call basic, okay? Now here you could either be having an excess of base, like hydroxide ions, or you're having low amounts of acid, so your pH is just going up, okay? Now these are just some basics. Now, an interesting formula that we've seen in chemistry before is that pH is equal to the negative log of hydrogen ions. So basically what they're saying is that the pH depends on the amount of hydrogen ions that you have. Okay, now why is pH important? We've talked about this a lot. Now pH basically regulates all the body functions, okay? There are enzymes and ion channels and hormones. All of these function at certain pHs, okay? And so you need to regulate this pH within a very narrow range in order to have the body functions working properly, okay? Now, throughout these body processes, okay, all the metabolism, you do get a lot of acids and bases produced, right? So it makes sense that these would keep on altering the pH. However, the body has very, very tight mechanisms in order to keep the pH levels constricted within a very narrow range, okay? Now, what do we use for this? We use what we call buffering systems, okay? All right, now let's take this figure. Now look at this formula up here, okay? We have water, and that reacts with carbon dioxide. Now, carbon dioxide is a waste product. We know that already. It's a waste product of tissues. All right. It reacts with water, and then it produces hydrogen ions and HCO3 minus. Now, HCO3 minus, we're going to be talking about this one a lot throughout this lecture. This is bicarbonate. Okay. So if I say bicarbonate or HCO3 minus, I'm talking about the same thing. Okay. All right. So how can we get rid of these substances? Now, we know that carbon dioxide, because it produces H plus, that means that it contributes to more acidity. Okay, so how can we get rid of carbon dioxide? Simple. Through the lungs, okay? You exhale your carbon dioxide and then that goes out into the air and then you have less carbon dioxide in your blood. What about hydrogen ions or even bicarbonate? How can you get rid of that? Of course, you can't exhale these ions, so there's a different way. You can go through the renal pathway. You can excrete these substances in the urine. Okay? Now, so basically, you will excrete your hydrogen ions or the bicarbonate throughout the urine. Okay, so aren't these enough? What do you guys think? Is it enough to excrete your carbon dioxide and your H plus ions or different ions through the lungs or the urine? If it's enough, I'd say that we don't need to talk about this lecture at all. Exactly, it's not enough. Okay? This whole lecture would be pointless if these mechanisms were enough. So let's talk a bit about what the formula we just saw. So again, CO2 reacts with water, produces bicarbonate and hydrogen, and it makes the blood more acidic. Okay? Now, carbon dioxide is eliminated in the lungs, and hydrogen, along with other acids and bases, is eliminated throughout the kidneys. Okay? So, like I told you, why do we need a buffering system then? So you guys answered that it's not enough, and that's correct. But why? Because in order to excrete these substances throughout the organs in the body, like the lungs and the kidneys, it takes so much time. And by the time that you've got rid of these substances, you've already changed the pH of your blood, and all the body functions would be distorted. And this is why we need buffers. Okay? All right. So what is an acid and what is a base? This is basic chemistry, okay? An acid is anything that dissociates into H plus and another molecule. Now this other molecule would be considered a base, all right? But basically anything that produces H plus. Now, what is a base? It's basically the opposite. It's anything that binds hydrogen ions. So basically, if you bind a hydrogen ion, this hydrogen ion is not going to change the pH and to become more acidic, and so we call this substance a base. Now these are formulas that show both an acid and a base. Now over here, you have HA. This is the acid. Now what do you see? You see that the reaction goes, let's take it to the right. It dissociates into H plus and A minus. Now this A minus is also considered a base. Why? Because if you take the reaction in the opposite direction, so to the left, this A minus will bind the H plus and produce a neutral molecule, okay? Now, for bases, we know that, again, hydroxide ions are one of the basic examples of what a base is. You see how it binds with hydrogen and produces water. Okay, so it basically neutralizes the hydrogen ion. This is what an acid and a base is. So now that we talked about those, then what is a buffer? Well, a buffer is actually a system of acid and base. So basically, when you see a formula and you have this HA, for example, this is the acid, and we said that how A minus is the base, then this pair of acid and base is what we call a buffer. What does this mean? It means that basically, if you add either acid or base throughout this equation, you can neutralize or alter the pH depending on what your change is. Okay, someone's asking when is this PAL session going to be uploaded? If it's today, honestly, I'm not sure. That's related to the team who's recording, so we can ask them at the end of the session. Okay? I'm not sure if it's today though. Okay. Now, back to the buffer. If we add an external acid, okay, let's say we add acid, and this acid starts producing H plus, okay? What's gonna happen? This A minus, which is the base, is gonna bind the acid, and it's going to neutralize it, okay? And so this way, you keep the pH in check, and it won't change. Now, the opposite happens if you add a base, okay? Let's say you add a base, and then it lowers the pH. What's this molecule going to do? This acid is going to dissociate to produce more hydrogen ions and to make the pH go back down so that you don't have this imbalance. Okay? All right, so here are some examples of buffers. Okay, on the left side you have the acids, and on the right side here you have the bases, and these are the hydrogen ions. Okay? Now here you can see that NH4 plus dissociates into NH3 and hydrogen. Now in the second equation, H3PO4 dissociates into H2PO4 minus, so the H went down by 1. Here you have 3, here you have 2, and the third one became the hydrogen ion. Now notice how here, this H2PO4 minus is the base, right? But look at the third equation. Here it's the acid, because this substance dissociates into HPO4 minus and H plus. So just because something was an acid in a specific context doesn't mean that it can be a base, okay? So if you want to say that this is an acid or a base, it depends on the context. It depends on what it's doing, okay? All right. Now, law of mass action. This is something that we're gonna briefly go through, okay? Now, you don't need to actually memorize how it's made, okay, or how to come up with this equation, and you're not gonna have to calculate anything using the equation, okay? This is just for understanding. So, law of mass action says that if you multiply the products of a reaction, now this is a chemical reaction, you have reactants, so HA, this acid is the reactant, and it produces A minus and H plus. So because these are the ones that are produced, we call these the products. So the law of mass action says that if you multiply the product, so H plus times A minus, and you divide that by your reactant, you always get a constant value, and we call that K. okay? Why is this important? Because if we alter this formula a bit, we get this thing, okay? I know it looks scary, but you do not have to use this to calculate anything, okay? You just have to remember what it looks like, but you're not gonna be asked to calculate any, all right? Now this equation is what we call a Henderson-Hasselbalch equation. Okay, it's a pretty weird name. Now what is this? This is basically the logarithmic form of the law of mass action. So basically they took the log of the previous formula that we saw, and then you get this. Okay. Now, why is this thing important? Because if you can see in the equation, we have pH and we have pK. Now, pK, this K relates to the constant value that we talked about, and you'll understand what it means later. Also, you can see here in the log, we have at the top A minus. Now, A minus is a base. And these two brackets like this means concentration. So if I put any molecule and I put these two brackets, I'm saying this is the concentration of this substance. So in other words, this ratio is the concentration of base over the concentration of acid. All right. So now you can start to guess that if we do any changes throughout the acid or base, this is going to change the entire result of the equation. Okay. All right. Now let's take the example. If pH is equal to pK of the system. Now when I say system, I mean the buffer system because we said K relates to the buffers. All right. Let's say pH equals pK. Okay. What does that mean? It means that if you take the difference of them and they're equal, the difference would be 0, right? In other words, what does that mean? Let's go back to simple math. If I take pK and I put it on the other side of the equation, it's going to be minus pK, right? So you're going to get pH minus pK, and we said that would be equal to 0. So what does that mean? That means that this log of concentration of base over acid also equals 0, all right? So this is what I'm saying. The log of the base over the acid must be equal to 0. Now from math, log of what number gives us 0? The log of 1, okay? In other words, the base must be equal to the acid, all right? So the concentration, this number over here, over this number, these two numbers should be the same so that if you divide them, let's say 5 and 5, you'd get 1. And now when you do log of 1, you get 0, okay? So what does this mean? What I'm saying is that the pH is equal to the pK of the buffer when you have 50% bases and 50% acids. In other words, the concentration of acids and bases are equal. All right? Now we'll go through this multiple times so you'll understand what this means. Okay, now let's extrapolate this formula, this Henderson-Hasselbalch equation, into a buffer curve. All right? Here on the x-axis, we've plotted the pH, okay, and each difference is basically related to the pK, so either just pK, pK plus 1, minus 1, and so on. On the y-axis, we have the base, okay, the concentration of base. Now, take this section over here, the middle, where we said pH equals pK. Remember how I told you this means acids and bases are equal? And that is correct, because let's go up all the way to the curve, okay? We stop here. And how much base do you have over here? You have 50%, okay? So you have 50% base, and the other 50% would be acid, and this is where pH equals pK. All right? Now, how do you go to this point, which is pK plus 1? Now, this means that if you go back to this formula, I'm saying pK plus 1. So the log of this ratio is equal to 1. Now, what number is that? Basically, you don't have to understand this like thoroughly, but for those who are interested, if we have 91% base, so I put 91 over here as the numerator. So what's 100 minus 91? You have 9. So 91% is base, 9% is acid. You get a number, and the log of that number is 1. So basically what I'm saying is that the concentration of base is going up. Now when the concentration of base is going up, you see here, your curve starts going upwards and to the right. If you go over here, this amounts to about 91%. And down here, it would be pK plus 1. For pK plus 2, the concentration of base goes even higher, up to 99%. And the same is true for the opposite direction. Okay? Now, why is this important? We'll see in a few slides. So again, talking about the buffer curve, I basically wrote everything that I said in text form. All right. Now, I think something else I wanted to add is, no, I think that's all. Okay. So basically everything I said is written over here. Okay. Now that we've seen the buffer curve, we want to talk about a concept called buffer capacity. Okay. So this basically measures how strong a buffer can basically bind the acid or base before the acid or base can change the pH. Okay. In other words, how good can a buffer neutralize an acid or a base that's added? Okay. So if you have, in other words, a high buffer capacity, then this means you have a good buffer. And the opposite, if you have a low buffer capacity, this means that you have a bad buffer. Okay. So how do we measure this? If you have a slight change in pH when you add a base or an acid, so you added a lot of base, for example, you expect the pH to go really high. However, you only had a very small change. This means that this was regulated very well, and so you have a high buffer capacity. Okay? However, if you get a big change in pH, so again, you added the same amount of base, but the pH changed dramatically, then this means that your buffer is not working well. Okay, so the buffer capacity depends on two things, and please remember these, okay? It'll make your life really easy. First thing is the pH and pK difference, and we'll talk about how this works in the next slide. Second thing is the total concentration of the buffer. Let's say that this buffer actually works well, okay? But you don't have much of it. Is it going to be able to regulate the acid and base? It won't, okay? So this is what we're talking. Okay. This is the same buffer curve that we looked at previously. All right. Now, notice how we added these red lines and this formula. Okay. Let's talk about the formula first. Buffer capacity. Again, we said this is how much a buffer can regulate the addition of an acid or base without changing the pH. So in other words, we're talking about the change of concentrations. So this triangle means delta. Delta means change. So the change in the concentration of base over the change in the pH value, generally. So let's take some numbers theoretically. Let's say I added about 10, I don't know, 10 millimoles of base, okay? And the change in pH was just 1, okay? This means that the buffer capacity is going to be 10 over 1, which is 10. So it's going to be pretty high, right? But let's say I added the same 10 millimoles of base. However, my pH changed about 5 points. So 10 divided by 5 would be 2. And this means the buffer capacity now is 2. 2 is much lower than 10. Okay, so this is the difference we're talking. Now let's see how this applies to this curve. See, notice how here the vertical lines, these 3 vertical lines, are basically the same percentage of base added. So we've added the same amounts of base in all 3 points, in pK, pK plus 1, and pK minus 1. Now the horizontal lines, these indicate the change in pH. Now I'm going to ask you, what do you notice about the change in pH along all these 3 points? There is one point that has a different change than the other 2. Did anyone notice something? Yes, the middle one has the least change, exactly. Okay, so pH at the point of pH equals PA. Correct. So what does this mean? This means that at this point, you have the best buffering capacity. So again, a buffer works best when its pK is equal to the pH of the solution it's in. Okay, so if I put a buffer and its pK is 3, what pH would work better? 3 or 9, for example? In which pH would the buffer work best? 3, okay? So this is what we're talking. So you see here that the change is very minimal. However, here at pK minus 1, the change in the pH was extreme. Also at pK plus 1, you see the change in the pH was extreme, okay? Notice something else about the curve. At this point where you guys correctly said that the buffer capacity is high, you see how the slope is very steep? Okay, this means that you're having a dramatic increase in the base value, but your pH is just changing a little bit. However, if you go to the other 2 points, the slope starts getting flatter, and so your change in pH is getting higher. Okay? Okay, so buffer capacity in the curve. Again, here I explained everything that we just talked about. Okay. Here. Now, here we said that this means that the highest buffering capacity of a buffer is when pH equals pK. Okay. However, everything in physiology has a range. So just because I said pH has to be equal to pK, in other words, the pH pK difference is 0, doesn't mean that if the difference is just 0.1, then that means the buffer won't work. No, there's a range. And what we want to keep the buffer in is a solution in which the pH and pK difference is not more than 1.3. Okay? And we'll see examples of that. But just keep the number 1.3 in your mind. Don't ask me why it's 1.3. I have no idea, okay? So again, remember how the buffering capacity depends on the pH-pK difference and the total buffer concentration. Okay? All right. So are there any questions so far? Anything that wasn't clear? I know I said the word buffer a lot. It's annoying. Okay, everything's clear. Great. Right. Now, let's take this example of three acid-base pairs. Okay, so three buffer systems. You have the HLA. HLA is basically lactic acid, and La minus is D-lactate. Okay, so basically it's lactic acid just in different forms. This middle one is the phosphate buffer, and this one is the ammonia buffer. Okay, so let's start with this one. Now, before that, let me orient you. So up here, you have the pK values of each buffering system. Here's 3.9, 6.8, and 9.4. Down here, I have the pH at which the solution is in. Okay, and here are the changes in either acid or base. Okay, but basically, we want to focus on the pK and pH. Now, let's take this first buffer, the lactate. Okay, the pK of this solution is 3.9. However, keep in mind that the pH of us humans is at about 7, okay? Now, there is a difference between the intracellular and extracellular pH. Intracellular, so inside the cells, is 6.8. Outside the cells, so basically in the blood, is 7.4. Now, remember this 7.4, we're going to use it a lot, okay? However, for this specific example, let's just stick to the number 7. So I told you the pH of our blood is 7. And the pK of this lactate buffer is 3.9. What is the pH-pK difference? Can you guys tell me? So what are you going to do? You're going to subtract pH and pK. So 3.1. Excellent. Okay, it is 3.1. So what did we say the value was that we should not exceed? 1.3. So 3.1 is definitely much higher than 1.3. So is this a good buffer? Excellent. Okay. All right, let's take the next example, which is the phosphate buffer. Okay, now the pK of this one is 6.8, and our pH is 7. So do you think that the phosphate buffer is a good buffer? Yes or no? Yes, it's actually an excellent buffer, and we use it a lot. Okay, all right. What about the ammonia buffer? So the pH in our blood is 7. The pK of ammonia is 9.4. So the difference is 2.4. Isn't that a high difference? It is. So is it a good buffer or not? It is not a good buffer. Excellent. Okay. So this take-home message is that as long as the pK is very close to our normal pH value, then it's a good buffer. Okay. So again, this is basically everything that I talked about. Okay, but you can quiz yourself again. Now, let's talk about the phosphate buffer itself. Okay. This is the buffer. We talked about this one earlier. Okay. Sorry. Do we need to memorize the values for each buffer? No, you don't need to memorize. You just need to know which buffers work well, and I listed them in a few slides. So just know that these buffers work well Anything else doesn't work. Okay, we don't need to memorize the pK for each one. You're welcome. Okay, so this is the phosphate buffer. H2PO4 minus dissociates into H plus and HPO4 2 minus. So you have 2 minus signs. Okay, we said its pK is 6.8. Okay, it's an excellent buffer for both the intracellular and extracellular compartments. Now, why am I separating intracellular and extracellular? Because we said the pHs are slightly different. We said that pH of the intracellular space is 6.8. Now, what's the pH-pK difference in this case? It would be 6.8 minus the intracellular pH of 6.8. It's 0. This means that it's an excellent buffer. Okay? What about extracellular? It's still good because the extracellular pH is 7.4 and the pK is 6.8. The difference is just 0.6. It's less than 1.3, so we're still pretty good. Okay. But remember I told you that there's another important factor for determining if the buffer is good or not? Something other than the pH-pK difference. What was it? The concentration of the buffer. Exactly. Okay. So if you have a high concentration of phosphate buffer, it's going to work well. So the question is the concentration of the phosphate high inside the cells? Yes, it is. Okay, I'm not going to expect you to know this. So yes, concentration inside the cells is high. However, is it high outside the cells in the blood? No. So can the phosphate buffer act well extracellularly? It cannot, okay? Because the concentration is low, all right? So conclusion, phosphate buffer only works intracellularly. And this is why we need other buffers to help us in the extracellular space or basically in the blood. And that is the bicarbonate CO2 buffer, okay? And we're gonna focus a lot on this one. So again, this is the formula. This is the regular formula that we've been talking about. Carbon dioxide with water produces H plus and bicarbonate. Okay, this is bicarbonate. Now, the pK of this system is 6.1. Don't tell me where I got this value from. I have no idea. Okay, now let's check the pH-pK difference both intracellularly and extracellularly. Okay? All right. So the pH intracellularly is 6.8, and we said the pK of the system is 6.1. The difference is 0.7. So this means that it is an excellent buffer inside the cells. Okay? And it also has high concentrations inside the cells. What about extracellular? We said extracellular pH is 7.4, and the pK here is 6.1. This means that the difference is 1.3. Now this isn't good, right? This means that this buffer doesn't work. But I was just saying a few minutes ago that this buffer is actually excellent and we depend on this one. How is that? Any wild guesses as to why we still use this bicarbonate carbon dioxide buffer, even though I'm telling you that the pH-pK difference is high? Any wild guesses as to why that's happening? We exhale the CO2. Excellent. Okay, so this means that the system is open. Other factors are contributing, okay? So you're having the carbon dioxide going outside, all right? So this means that you cannot determine and say that the pH-pK difference basically guards or says that you have to stick to 1.3, okay? It's fine because it's an open system. Carbon dioxide is exhaled. So this whole system is dynamic. So it just changes based on what we need and based on how the pH in the blood is, okay? So again, the pH-pK difference does not affect the system, and the whole balance is always going to be shifted towards the left side. Now, why is that? Remember from chemistry, if something is always excreted out, then the whole equation would be attracted to go to that side to make more of this. So in other words, carbon dioxide is always exhaled, so these two would always want to make more carbon dioxide. All right. Now, obviously, this is different in cases if we have high carbon dioxide in the body, and we'll see those later on. So, conclusion, this buffer, bicarbonate and CO2, works excellently both intra and extracellularly in the blood. Okay, so we talked about the phosphate buffer, the bicarbonate buffer, but we also need other buffers. The third example is protein buffers. okay? Now, obviously, you don't have to memorize this thing, the structure, the names, no need for that, okay? What I'm trying to show you here is that you have this whole protein, this peptide chain, you have the carboxyl end, and you have the amino end over here, and you have different amino acids, okay? Now, what do you notice? You notice a lot of hydrogen is being taken in and out, which means that all of these are buffers on their own, okay? Now the question is, which one of these amino acids is a good buffer and has a pK that's close to our pH? So I'll show you this figure, okay? Now our pH is about 7, right? You can see that these three substances, so the histidine amino acid, the cysteine amino acid, and the amino group at the end, these all have a pK that's close to 7. So this means that these function properly. So you can see here, we've put them in red, the ammonia group at the end, the cysteine amino acid, and the histidine amino acid. These three are excellent buffers. And I can see, however, that the others are not good buffers for our body, okay? However, a general rule is that proteins are excellent buffers generally. Why? Because they cover a wide range of pH. Okay, so if you basically put a protein in any solution with any pH, it's going to be able to buffer excellently because there's so many amino acids and each one has a different pH. Okay, so this is basically everything that I said. All right. So we finished talking about the three types of buffers, but we're gonna focus a bit on the bicarbonate carbon dioxide buffer. Okay, I just want to make sure that everything's clear so far before we dig into this one. Is everything clear? Yes, okay, excellent. Is this the second lecture? We're actually still in the first lecture. Yeah. Okay. But this is the boring part. I understand. However, we're gonna get to the other one pretty soon. Okay. Now let's talk about the bicarbonate and carbon dioxide buffer. All right. Now we want to talk about it in terms of the Henderson-Hasselbalch equation, as you can see here. You can see that the pH is about 7.4. This is the normal pH in our blood. Now the pK we said of the system is 6.1. Plus the log, now we said log would be of base over acid, sorry. So the base in this case is the bicarbonate, and the acid is the carbon dioxide. Now, we know that from labs or whatever, that the normal value of bicarbonate is 24. And we know that the PCO2, which is the partial pressure of carbon dioxide in the body, in the blood, is 40. Okay? So where did we get this 1.3? Well, we got it from basically a formula. Now, you don't need to memorize the formula, but it's just saying that the concentration of CO2, which is this one, equals the PCO2, which is 40, times a constant. Okay? Now, this constant happens to be 0. Okay, this constant happens to be 0.03, so 40 times 0.03 is 1.2 millimoles, and that's how they got this value. Okay, however, you don't really need to use 1.2. We don't usually use that. And when we talk about the disturbances, we look at the PCO2 of 40. We don't really look at 1.2. Okay, let's extrapolate this equation onto the graph. So you get this, okay? Now this is what we call a Davenport diagram, which is basically a plot of pH against the bicarbonate, and you have these lines, okay? Now let's take this one, the normal one, which is PCO2 of 40. Okay, this is a normal. Now you can see that any change along this line occurs along the same partial pressure of carbon dioxide. So we call this an isobar. Iso means same, and bar means pressure. So what I'm telling you is any change across this line is happening along the same pressure of carbon dioxide, which is 40. Okay, I can see here how as you go to the left, the pH is decreasing and your bicarbonate is decreasing. Okay, so bicarbonate is a base, so if it decreases, it makes sense that your pH would go down. You have more acidic, right? Now, we have two other isobars. We have isobar 20, isobar 60, okay? The point I want to show you here is that when you have a lower concentration of carbon dioxide generally, okay, your bicarbonate also goes down. And if you have a higher concentration of CO2, again, your bicarbonate goes up. Okay, so basically these are trying to balance out each other. We're gonna go through this type of diagram a lot, like soon. Okay, so again, the Davenport diagram, it's important to understand it because when you understand it, you're gonna know how to basically see the compensations, the disturbances, and all of that. So the middle slope is the isobar of 40, which means that the pressure of carbon dioxide is 40 along all the points. The topmost line was isobar 60, the lower one was 20. And notice how, with each line, as the pH increases, the bicarbonate concentration increases as well, because bicarbonate is a base, so if it increases, the pH would also go up, and vice versa. Now, when PCO2 is 60 compared to 40, so we have a higher PCO2, the overall concentration of bicarbonate is higher, okay? And this is to compensate for the increased acidity that you're getting. Okay, now what happens if the bicarbonate CO2 buffer was the only buffer in the body? Now, I did give you a hint a while ago that this buffer is not enough and you need others. But let's say theoretically, what happens if this is the only buffer in the body? So, remember how we have the hydrogen and the bicarbonates. Okay, these are the reactants, sorry, the products. So we have them over here. Hydrogen times bicarbonate over CO2 gives you a constant value of K. Now, let's take a scenario. If you have one molecule of CO2 in your body, okay? Obviously, that's not true, but theoretically, let's say you have one molecule of CO2 in your body over here. And let's say you decided to stop breathing for whatever reason, okay? You're tired of it. Okay, I'm going to stop breathing. Theoretically, you would get an increase in your carbon dioxide, okay? Let's say that increase was also one more molecule. So now your carbon dioxide went from just one molecule to two molecules. So what happened? You got a doubling of your carbon dioxide, okay? Your carbon dioxide doubled, so basically over here, this number doubled. Now we want to keep this value constant, right? So what do we have to do to these two up here? We also have to double them in order to make sure that the K value remains the same. So basically what I'm saying is that any change that happens to the denominator should also happen in the numerator, okay? And this is from math. Okay, so how do we do that? Now, here are some crazy numbers. The bicarbonate concentration in the body generally is 600,000 times more than the concentration of H+. So let's say theoretically that you have 600,000 molecules of bicarbonate in the body and you'd only have one molecule of H+. Okay? Now, we said that we added one molecule of carbon dioxide to the system. Okay? If you add one molecule of carbon dioxide to the system, it's going to go through this reaction and produce what? One bicarb and one hydrogen. Okay, so then you're adding one, so you're getting one of each in the reaction. What does this mean to us? It means that to the 600,000 molecules of bicarbonate, you just added one bicarbonate. Now, to you, does that make a difference? Are these two numbers different than each other? No, okay, it's a very minimal change. However, for H+, we said that it was just one molecule. And now we added a second one. So did this change anything? Yes, the value of hydrogen also doubled, okay? And this is why we say that when you add CO2, you increase the acidity because you're mainly changing the hydrogen. So basically the ratio or the basically relativity of increasing the hydrogen is much more than the bicarbonate. And now, let's say you doubled the hydrogen now. So from chemistry, if you guys remember, if you have a lot of products, the reaction becomes what we call saturated, and it will not want to continue producing more, right? So this means that your buffer did not work properly. Why? Because you had a dramatic increase in hydrogen when the carbon dioxide went up. And obviously, if your hydrogen doubled the number, that means your pH is going to change dramatically. And this is why we cannot use this buffer on its own. Okay, why am I going through all this? Because I'm showing you that you cannot use this buffer on its own. This buffer on its own cannot regulate the hydrogen, and you're just going to get a lot of hydrogen being produced. Okay, so this is the Davenport diagram in the case of having only bicarbonate. So the bicarbonate CO2 buffer. So this would be what we call the buffer line. This is a red line, okay? And this shows basically the changes that happen when you have changes in CO2, okay? You notice how over here it says hemoglobin is 0. Now what is hemoglobin? It's a protein, right? And we said proteins are buffers. Now what I'm telling you is that in this system, we don't have proteins. So your hemoglobin is technically 0. This obviously won't happen. If someone has this, then they're probably dead. Okay? But theoretically speaking, let's say you don't have any other buffers unless this buffer that we talked about. Okay? You notice how the bicarbonate concentration is always over here. Remember I said the normal value is 24, so it's always at 24. Is it changing when I'm changing the carbon dioxide? So I'm going from this isobar of 40 to, for example, the isobar of 80. And we said the change happens along the red line. So I'm going from this point to this point. Are you seeing any change in the bicarbonate? Can you guys tell me? Do you see any change in the bicarbonate? No, there's no change in the bicarbonate. Okay? And so this is what I'm trying to tell you. That the bicarbonate is not even trying to compensate, right? Alright. What about the pH? You see that the pH is dramatically decreasing. Okay? It's becoming very acidic. So this means that this buffer on its own cannot regulate any disturbance. Okay? Alright. So this is basically everything that I said. What message is that this shows the importance of the other buffer systems, and thankfully we do have these systems, okay? Alright, now let's see what normally happens in the human body when we have other buffers, alright? So, the other buffers are represented in this equation, that H plus B minus gives you HB. So H plus is the hydrogen, B minus is the base, and these two combine to produce HB, which is a neutral molecule. Now let's go theoretically through the same things that we went through. Let's say you increase CO2 again because you decided that you're fed up with breathing, you don't want to breathe anymore. Your CO2 goes up, okay? This reacts with water and produces bicarbonate and hydrogen. Now, we know that in the previous case, hydrogen will stop the reaction because you produce too much hydrogen and it's not being neutralized by anything else. However, in this case, the base is going to bind the hydrogen, and so you will get a production of HB, okay? This means what? Now I'm taking up the hydrogen. So I'm basically emptying the room for carbon dioxide to react with water and produce more hydrogen, okay? And so the cycle keeps on repeating itself. So what am I getting in the end? In the end, I'm getting an increase in my bicarbonate, but I'm also getting a decrease in the other bases, okay? Also, I'm getting an increase in this neutral molecule. So in other words, am I getting any change in bases? No, because any increase that happens with my bicarbonate, the B minus, the decrease in B minus, basically compensates for that. Okay? Now you'll tell me, okay, if that happens, then you're not getting any change, right? Because you're balancing the loss and gain of bases and you're taking up the hydrogen. However, keep in mind that sorry, I think I missed that point. Okay. Keep in mind that when you have this reaction, there's always a hydrogen ion that escapes. Okay? There's always a hydrogen ion that's sneaking and it goes away and it doesn't get taken up by the other bases. And so that's why this extra hydrogen ion is going to go away and it's going to lower the pH. Okay? It's going to make the blood more acidic. And this is how carbon dioxide basically makes the blood acidic. And we're going to go through this later. Okay, so again, this is the same buffer if we only have bicarbonates and carbon dioxide without any other proteins. You see the bicarbonate doesn't change. However, in the case of normal, like our normal levels, hemoglobin normally is about 15, okay? So you can see that as your PCO2 changes, let's take 40, the isobar of 40, and let's say you go down to 20. What do you see? You see a decrease in the bicarbonate, right? And this is a normal reaction that you would expect, okay? Again, same thing. If you went from PCO2 of 40 up to 80, what happens? Your bicarbonate goes up to try to compensate for that, okay? Now let's say, for example, that a person has anemia, okay? And their hemoglobin goes down to 5, theoretically speaking. Now you can see that the changes in hemoglobin are not much, right? This is because, sorry, the changes in bicarbonates are not high enough. Why is that? This is because you're having less amount of protein. Remember, we said hemoglobin is a protein, and this is a protein that you find in red blood cells. Okay, so red blood cells are basically like a bag of hemoglobin, all right? And this hemoglobin, essentially, it's a protein, so it acts as a really good buffer. So if someone has anemia, this means that they have low hemoglobin. So they have low levels of this protein buffer. And so that's why your bicarbonate would not change as much and it would not be able to compensate as much as normal. Now let's take the opposite direction. Okay, if you have polycythemia, in which basically your hemoglobin goes really high up to 20, for example, you see that this line became very steep. So even a small change in carbon dioxide is going to dramatically increase or decrease your bicarbonate. Okay, was this part clear about the bicarbonate and carbon dioxide buffer? Okay, excellent. All right. So now we can talk about some normal metabolism. Okay, now let's see what happens throughout the body. Remember I told you at the beginning that we have throughout body processes, we produce acids and bases. So now we're going to talk about some of these substances. So how do we produce carbon dioxide generally? We know it's produced throughout all body tissues. But how do we do that? So you have this molecule, C6H12O6. This is glucose. Now when glucose reacts with oxygen, it produces carbon dioxide and water and obviously ADP. But that's not our point. Our point is the CO2. Okay? Now, the total amount of CO2 produced in our bodies is 20,000 moles per day. Now, where is this excreted? I asked you guys this question before, and you guys said throughout the lungs. Okay? Now let's talk more about that excretion. Now, we know that carbon dioxide, again, is produced in all body tissues. Now, this carbon dioxide would diffuse out from the tissues of the body to the blood, okay? And this basically happens in the veins, okay? So the carbon dioxide would be pushed out of the tissue into the veins and into the venous blood, okay? Now, this venous blood, where does it go? It goes to the heart, and then it's pumped to the lungs for what? Oxygenation. Okay? Pretty straightforward. Now, what happens when the blood reaches the lungs? The carbon dioxide is basically going to diffuse out of the blood into the alveoli and is going to be exhaled. Now, did you notice how we never mentioned that carbon dioxide is in the arterial blood? That is because we said it's always pushed out into the venous side. Now, this doesn't mean that I'm telling you the arterial blood does not have carbon dioxide. No, of course it does. But I'm just saying that the carbon dioxide in the venous side is much higher than the arterial side. Okay? And so this means that the venous blood is more acidic than the arterial blood. And so you have a form of respiratory acidosis. Okay? And this is the first time we talk about a type of disturbance, but this is normal, okay? Respiratory means that it's coming from the lungs, it's coming from carbon dioxide. Metabolic means it's coming from the body, from the kidneys, and so in this case, we would usually look at the bicarbonate. Now we're going to talk more about that later. Okay, we saw where hydrogen and hydroxide, we saw where carbon dioxide comes from. Now we're going to see where hydrogen and hydroxide ions come from. So, I'm going to ask you this. Do these two ions come from carbohydrate metabolism? What do you guys think? I just gave you an example of metabolism of glucose. So no, okay? You do not get these two ions from carbohydrate metabolism. You only get carbon dioxide. What about fat metabolism? What do you think? You think fats would produce hydrogen and hydroxide ions when metabolized? Okay, no again. Right. So it basically just produces carbon dioxide. So what is the last macromolecule we have left that would produce hydrogen and hydroxide? We said carbohydrates, no. Fats, no. And proteins. Excellent. Okay, so amino acid metabolism would produce hydrogen and hydroxide ions. Now, because this comes from proteins, so it depends on what ultimately the food that you eat. So if you eat more proteins, you'll have more production of the hydrogen and hydroxide ions. Okay, and so now we're going to talk about the amino acids. So protein metabolism, we said it produces both hydrogen and hydroxide. Now, unfortunately, this is a kind of annoying memorization part. You have to know which amino acid, like which types of amino acids produce hydrogen and which types produce hydroxide. Okay, so generally for hydrogen, it's the sulfur-containing amino acids like methionine, cysteine, and cysteine. Also, the cationic amino acids, so lysine, arginine, and histidine. For the hydroxide, you have the anionic amino acids like glutamate and aspartate, and the salts of organic acids like acetate and lactate. Okay. So how much hydrogen and hydroxide do we obtain? Now we said it depends on food, but let's say we all eat a standardized diet, okay? How much would we get? Now from the sulfur and cationic amino acids, we said these produce H+. And these actually produce about 210 millimoles of H+. Okay? For the anions and the salts, this produces about 130 millimoles of hydroxide. Now, keep in mind that hydrogen and hydroxide always neutralize each other into water. So if you have an equal amount, you wouldn't have any change in pH. However, is this amount equal from the food that we eat, or do you have an excess of something? So you basically have, yes, we do have an excess, okay? We have an excess of hydrogen, okay? We have actually 80 millimoles difference, okay? Because we said from the food you get 210 hydrogen and 130 hydroxide, okay? So again, we said that each molecule of hydrogen and hydroxide would neutralize each other and become water. So this means that these extra 80 ions must be excreted. And we know that the kidneys can excrete hydrogen through the urine. However, in order to be able to do that, you need to produce 2,667 liters of urine per day. Now, there is no way that any human can do that, okay? You could take it as a challenge, but there is absolutely no way that you could produce this amount of urine per day, okay? So, what does this mean? You need buffers for this, all right? Okay, let's talk about the excretion of hydrogen ions in the kidneys, okay? All right, so we have basically bicarbonate, and that is filtered throughout the glomerular capillaries. All right, so it's filtered from the blood into the lumen, okay, with the urine. However, we don't want to lose this bicarbonate. We're trying to excrete hydrogen, right? However, bicarbonate cannot cross back into the tubular cells because it is charged. So what do? How can we take it back? We take it back through carbon dioxide. Okay? Now, where do we get this carbon dioxide? Now, in the tubular cell, since it's a cell, it normally produces carbon dioxide. Okay? Now, this carbon dioxide would react with water and it would form hydrogen and bicarbonate. We've seen this reaction many times. And this is how the bicarbonate gets taken back into the blood. So this is kind of like a shuttle bus, okay? You push it out of the blood and then you take it back in through the tubular cells. Now, we said carbon dioxide is the way to go, but how do we get this carbon dioxide? Well, think about how the hydrogen is also filtered with the bicarbonate. Okay, so you also get hydrogen filtered out into the blood. This hydrogen would react, this hydrogen would react with the bicarbonate and it would form carbon dioxide and water. Again, same reaction as here, it's just outside in the lumen. Now, this carbon dioxide is able to diffuse into the cells because it is not charged. Okay? And this is where it's going to dissociate into hydrogen and bicarb would be taken up in the blood. Okay, so bicarbonate is taken up in the blood. All right, what about H plus? This is what we're trying to excrete, right? Well, there is a certain transporter that pushes the H plus back out into the lumen, right? However, this process is not enough to excrete all the hydrogen ions that are extra. Okay, we have 80 millimoles, and that's a pretty big amount. Okay, so we need other mechanisms. The second thing is the phosphate. Okay, so remember the phosphate buffer? This is the one we're talking about. Now, you have phosphates, and that is excreted out in the form of HPO4 minus. Okay, this reacts with hydrogen and it produces H2PO4 minus, and this one would be excreted in the urine, and this actually contributes to the acidity of the urine. Okay, however, what did we say about phosphates being an extracellular buffer? Does it work extracellularly? Remember, we said the pH-pK difference and the concentration. So because phosphate has a low concentration extracellularity, then it cannot work as an extracellular buffer in the urine. So again, we need something else to help. What is that? That is glutamine. Okay, this one over here. Now this is basically an amino acid, and we have it in tubular cells, and this one would dissociate into ammonia, so NH4 plus, and alpha ketoglutarate. Okay, and it was very random, it came out of nowhere, but this is the way we can effectively excrete hydrogen. Now, this NH4 plus cannot go outside of the cell because it's an acid, so okay, we can just excrete that. However, you can't cross the cell membrane because it is charged. So what do you need to do? You need to neutralize it initially. So you dissociate NH4 plus into NH3 and hydrogen. NH3 can readily go out into the lumen. However, H plus, it's charged, but it does have a transporter, so that pushes it out into the lumen. And again, NH4 plus is gonna be formed because NH3 and H plus, they can't stay away from each other, they always want to be together. And so you'll have NH4 plus being produced again. And then this goes out into the urine. And actually, this ammonia is what gives urine this characteristic smell. Okay, especially if urine stays for a while in a specific area, then that is gonna make a bad smell, and that comes from the ammonia. NH3 is not charged, it actually isn't. Okay, it's just NH4 plus. Okay, so NH4 plus, and then you take the H plus out, so you're left with an NH3. Okay, NH3 is not charged, and so it can cross the membrane without needing any transporting. Oh, I wrote it here. Okay, I'll fix that. Thank you for pointing that out. I wrote NH3 plus here. This is just supposed to be NH3. Okay, now we said, all right, we've excreted the H plus. However, we have alpha ketoglutarate left here. It feels lonely, okay? So we gotta get that back inside the blood. So how can we do that? Basically, remember this formula again? The carbon dioxide with water reacts to form hydrogen and bicarb. So we're gonna need this hydrogen, okay? Now this hydrogen from this reaction would react with alpha ketoglutarate and would produce glucose. And now glucose is just going to be readily absorbed into the blood. Okay? But notice how we took up a hydrogen, but at the same time, we also produced a bicarbonate. And this bicarbonate gets taken up into the blood. However, this bicarbonate is new. Okay? So we didn't get this bicarbonate from what we got from the blood initially. This is just produced on its own from carbon dioxide. Okay? So this means that for each molecule of hydrogen that you secrete, you produce a molecule of bicarbonate. Okay? Did you guys get this point? And this is how we can help our body balance the pH because we have a lot of hydrogen. We push that out, and at the same time, we're making new bicarbonates in the kidneys and taking it out. Okay? Okay, you'd like me to repeat? Okay, I'll repeat. Okay. So in the slide, I was saying that we have alpha ketoglutarate. It's produced from the dissociation of glutamine into ammonia and alpha ketoglutarate. Okay? Now, alpha ketoglutarate doesn't just want to stay there in the cell, it wants to be taken up again. But it can't be doing that on its own. It has to be basically it has to go into glucose. Now, how does it do that? It needs hydrogen. So you need alpha ketoglutarate to combine with hydrogen to produce glucose. Okay? And then the glucose would be taken up. Now, the point here that's different is that where do we get this hydrogen from? We get it from the same reaction that we had before, okay? The carbon dioxide with water would produce H plus and HCO3 minus, okay? However, this reaction, okay, it's not coming from substances that came through the blood. It's not coming from the bicarbonates that we initially filtered out. This is just coming from carbon dioxide that was in the cell, and then it's just dissociating into hydrogen and bicarbonates. Now, the hydrogen is being neutralized with alpha ketoglutarate and it becomes glucose. So what is the next thing? You have bicarbonate. And bicarbonate is not bound to anything to neutralize it. So this base just goes into the blood. So what does this mean? I'm excreting hydrogen, so I'm making the blood less acidic, and I'm also taking up more base. I'm taking up more and new molecules of bicarbonate. Did you understand this part? I know this is probably the most confusing part of the whole lecture, actually. So you understood it? Okay, great. Okay, so now we are done with half of the material. Now we're going to talk about the acid-base disturbances. However, we could take a 5-minute break if you guys want to, or we could just go ahead and continue. What do you guys prefer? If you guys want to take a break, I don't mind. If you want to continue, I don't mind. Okay. So no one's saying to stop, so I'm just gonna go ahead and continue. Okay. All right. We can continue. Okay, let's continue. So now we're going to talk about the acid-base disturbances. Okay. All right. So there are specific parameters that we need to follow, and basically these are certain benchmarks. You're going to use these, and you're going to look at it. If it's acidosis, alkalosis, and then you're just going to look at each one, and this is going to help you determine what kind of disturbance you're having. So first thing is pH. Again, pH determines if we are in a state of acidosis or alkalosis. Okay? Now, a low pH means acidosis, and this is a nice mnemonic I found, sliding down the pH down, okay? For alkalosis, your pH is going up, so K in alkalosis means kicking the pH up. Now keep in mind that even though we said that pH relies on the hydrogen, don't look at the value of hydrogen alone. So if you take a patient's blood test, don't look at the hydrogen. Just because the hydrogen is high doesn't mean that the patient's having acidosis. Okay? The patient could be having alkalosis, but the pH is high for whatever other reason. Okay? Now, the normal value of pH is 7.35 to 7.45. Okay. Now, we know acidosis versus alkalosis. Now, when do you say it's respiratory and when do you say it's metabolic? Respiratory, either acidosis or alkalosis, means that it's coming from the respiration, from breathing. Okay? So this case means that the parameter would be CO2, carbon dioxide. Okay, carbon dioxide is highly affected by the rate of breathing. Okay, so if we start breathing less, okay, what's gonna happen? Am I gonna excrete or exhale more carbon dioxide? No, my carbon dioxide is gonna accumulate in my lungs and in my blood, and this will give me respiratory acidosis. Now, if the ventilation increases, meaning that I start breathing rapidly, I'm exhaling more and more carbon dioxide. And so this gives me a form of respiratory alkalosis. Now, what about metabolic? Metabolic also means non-respiratory. That's coming from other causes. It can be the kidney, it can be the body's metabolisms, whatever. But it's not coming from respiration. So the parameter in this case would be what? What do you guys expect the parameter for metabolic disturbances to be? We said respiratory is carbon dioxide. What would metabolic be? HCO3, excellent. Okay, it would be bicarbonate. Now, you're right, bicarbonate is the main one, but we have two other things, okay? For the bicarbonate, we use the standard value of 24. Now, that's the one we benchmark against. We don't test each patient's actual bicarbonates, and then we see, oh, is this normal relative to the patient? No, we always compare it to 24. The two other parameters are buffer bases and base X's. Again, new terms, what is going on? We'll go through that. So buffer bases, okay? Let's take this reaction. We've seen it a bazillion times, and we're going to go through it again. So buffer bases are basically the total concentration of bases in the blood. Okay, now what are the bases over here? These are bicarbonates and the B minus, the other bases. Okay, again, carbon dioxide and water produce bicarbonates and hydrogen. Bicarbonate is a base, and then you also have other buffers that act as bases to neutralize the acid. These two bases make up the buffer bases, which is basically the total concentration of bases. Now, let's consider this respiratory change. You have an increase in your CO2. What's going to happen? Each increase in CO2 is going to make one bicarbonate and one H plus. Now, even though the H plus increases by 1, okay, the B minus would decrease because it's going to bind this H plus and neutralize it. Now, does this mean that we have more base because we're basically neutralizing the hydrogen and we're having an increase in bicarbonates? What do you guys think? So we said that we have a dissociation. You have bicarbonates and hydrogen. The hydrogen is being taken up by the B minus. So does this mean that we're having more base and we're going into alkalosis? What do you guys think? We have an increase in bicarbonates and a decrease in B minus. So no, exactly, okay? So basically, for each bicarbonate that's produced, you have one B minus that's taken up, and so they cancel out. So you don't go into alkalosis just because you're producing bicarbonate. So the sum of buffer bases, basically the sum that we talked about, remains the same. So we just increase this by 1, decrease this by 1, they cancel out each other. So this means that the buffer basis does not depend on the CO2 changes. So if my disturbance is coming from a change in carbon dioxide, my buffer bases are not going to change, okay? And so this is why this is a parameter of metabolic changes, not respiratory. Okay, now we solved the dilemma of the blood becoming more basic. We said that no, because bicarb and B minus balance out each other. Now what about hydrogen? We said it's taken up, right? However, there's always one molecule of hydrogen that escapes and changes the pH. Okay, so one molecule after one molecule, this is going to change the pH. Okay. Now, let's take the case of a metabolic change. We talked about respiratory change. Let's go for metabolic. Let's say the pH, the H plus, sorry, the hydrogen increases. Okay? What's going to happen? This hydrogen is going to be taken up or bound by both the bicarbonates and the B minus to try to neutralize it. Correct? So react with bicarbonate to produce carbon dioxide. However, the carbon dioxide is not going to accumulate again because it's exhaled through the lungs. So you get a decrease in your bicarbonates. You also get a decrease in B minus because the B minus binds the extra H plus and it tries to neutralize it into HB. So what happens to your buffer bases, which is the sum of bases, which is the sum of bicarbonates and B minus? What happens to that sum? Does it increase? Does it decrease? Does it stay the same? It decreases. Okay? And now that it decreases, what does this mean? This means we're going into acidosis because your basis, the total amount of bases in the blood, is going down. Okay? Now that you understand buffer bases, you'll definitely understand what base excess is. Okay? Now, base excess is basically the difference between the actual buffer bases in a person and the normal buffer basis value. Okay? And the normal buffer basis value is about 48, and the actual is what you test in a patient. Now, this is the parameter that according to the doctor, this is the one that's used the most in the clinics, and it's the most helpful and accurate one. However, from what I've seen, we use bicarbonates. Okay? Alright, now the normal value of base excess is -3 to +3. Okay, even though we usually say it's 0, like 0 is a normal value, which means that buffer bases are equal to 48. However, this small range still means that it's normal. Now, if the value is higher than plus 3, this means that you're having an excess in your bases that's more than normal, and so you're having alkalosis because your number of bases are high. And the opposite is true. If it's lower than 3, you have less bases, so you're going into acidosis. Now, if the actual buffer base equals the normal buffer base, which is 48, this means that your base excess is 0, and we set that. Now, this is just a table showing you the normal values. Now, you don't have to memorize the ranges, okay? The only things you would have to memorize are the average values of everything, okay? So partial CO2 pressure is 40, we know that. pH is 7.4, sorry, in blend. However, know that the normal range is 7.35 to 7.45. For bicarbonates, you have 24, and base excess is usually 0, but the range is -3 to +3, and your buffer bases should be about 48. Okay, we got that out of the way. Now we're actually going to start talking about the disturbances, so acidosis and alkalosis. Acidosis can come from either a respiratory cause or a metabolic cause. Respiratory means that it's coming from PCO2, it's coming from carbon dioxide. Metabolic means that it's coming from hydrogen or hydroxide. Okay, basically you're getting changes in the metabolic things, the ions. And the same thing goes for alkalosis. Now this is another diagram that I think is very helpful. Okay, respiratory comes from the lungs, metabolic comes from the kidneys or other things. Okay, now acidosis means that your pH is going down because you're having an increase in carbon dioxide for respiratory. Alkalosis is the opposite, and same for acidosis. Acidosis means your pH is going down because your bicarbonate is decreasing. So you're having less base, so your blood is becoming more acidic. And the opposite is true for alkalosis. Okay, let's take the example of respiratory acidosis. Okay, this is a famous one. They love asking about acidosis, by the way, more than alkalosis. Alright, so focus on respiratory and metabolic acidosis. Now let's take the case of respiratory acidosis. In respiratory acidosis, what is the primary disturbance that you guys expect? So from the name, respiratory acidosis, what's happening? Exactly. You're having an increase in carbon dioxide. Okay? So this is your primary change. Now what happens? The reaction will be going that side. So this increased carbon dioxide will bind water, will produce bicarbonate and H plus. Now this H plus is going to be taken up by B minus and it will produce HB. Okay? So again, we mentioned this before. Any increase in HCO3 is balanced by a decrease in B minus. So your buffer bases would be the same. It would be equal. Base excess would be 0. And your pH goes down. So this is what we're saying. Okay? Now this is everything that we said. Alright? However, remember that there are always some hydrogen ions that escape and they make the pH go down. Okay? So PCO2 goes up, pH goes down, bicarbonate increases, but that's balanced by a decrease in the B minus. So these two are equal. The buffer bases are normal still, and your base excess is 0. So now because of this hydrogen that keeps on escaping, you need a way to compensate this increased hydrogen. Now, I want to ask you guys, what do you think is a way in which we can compensate for this disturbance? So the reason we have acidosis is because of the H plus? Yes, okay? The reason why you have this respiratory acidosis, I know that we said the primary disturbance is carbon dioxide, but in the end, this produces what? Hydrogen ions. And these hydrogen ions are acidic and they make the pH go down. However, your primary disturbance is carbon dioxide. So now that we know this, what do you guys expect to be the compensation in this case? How can we balance this? We produce base to compensate. Excellent. So what do you want to do? You want to increase the bicarbonate. Now, I know we said that the bicarbonate increases because you're producing it from CO2, but it doesn't increase as much. It doesn't increase enough to compensate. Okay? Now, before moving on to that, let's talk about the Henderson-Hasselbalch equation again, so you guys understand the ratios. So, We said that the primary disturbance is that CO2 increases, and that is the denominator in this ratio. Now, the bicarbonate also increases, but it's not enough. Okay, it's not enough to compensate this. This just happens as a reaction. All right. Now, here you have a small number, and here you have a big number. Small divided by big is what? So, okay. And so 6.1 would be added to a smaller number. And that's why your pH goes down. Okay? All right. So for this case, you need a compensation. Now remember, respiratory problems are compensated through metabolic pathways. What does this mean? You guys already understood this concept, but let me clarify. So if your initial problem is coming from a respiratory issue, so it's coming from carbon dioxide, you cannot say, okay, I'm going to compensate by changing the carbon dioxide, because that is the primary disturbance. So you're going to have to use the other pathway, which is the metabolic pathway. In this case, it's bicarbonate. Same as for the other side. So if you have metabolic disturbance, your bicarbonate is the one that's changing. You cannot say, okay, I'm going to change my bicarbonate. No, in this case, you would change your carbon dioxide. Okay? All right. So this is the compensation that we got. Okay? The kidneys start to increase the bicarbonate, and this would make the blood less acidic. So you see the green arrow over here. So you have an increase in bicarbonates. And now, because you added more bicarbonates to the buffer basis, then the value would increase. Okay? And now the base excess, because you're adding more base, it would be more than 0 and more than 1. Okay? Now, what type of compensation is this? All right? I know it's written, and so I won't ask you. The type of compensation is metabolic alkalosis. Now, what do I mean by that? I mean that in order to compensate, the body does what would happen in a state of metabolic alkalosis. Metabolic means it's coming from bicarbonate. And alkalosis means that the pH is going up, so you're having an increase in your bicarbonate. This is the compensation. Okay, now let's take this and extrapolate all that we said onto a Davenport diagram. Okay, so this is the normal buffer line. Okay, now keep in mind any change that happens along the buffer line is a respiratory problem. So you're having this red circle going to this blue circle. So what's happening to the pH is going down. Okay, and you see that the bicarbonate increases. But is this the compensation? No, it's not. Okay, it just increases just a little bit. Okay, but you're still going into severe acidosis. All right, so keep in mind that in this case, we moved from isobar 40 to isobar 60 or 80. Okay, point is that I increased my carbon dioxide. Okay, and in that case, this circle goes along the buffer line. All right, now this green arrow is the compensation. How can we compensate? We're going to go along the same isobar, so we're still at CO2 of 60. However, I'm changing what my bicarbonate concentration. And what am I doing to it? I am increasing it. Okay? So you can see that while I'm increasing it, it is going towards the right side. And so what happens to the pH? At first I was at 7.2, and now I'm about 7.3. So I'm compensating, right? So this is basically what I said over here. Okay? However, you did not reach this normal pH line. Now this dotted line is important. This signifies the normal pH of 7.4. Now did we reach it with this compensation? No, we didn't. Okay? So this means that you're going to still have to increase your bicarbonate much more until you reach this dotted line. Once you reach this dotted line, your pH is normal, and now all the levels of carbon dioxide and bicarbonate start to go down, and you reach a normal state again. Okay? Now, keep in mind, or actually, let me ask you, do you think that the body can overcompensate? So now, this is a case of respiratory acidosis. Do you think that because, for example, I keep on increasing my bicarbonate, this circle is going to go all the way over here and into alkalosis? Do you think that's possible? It is not possible. Okay? The body never overcompensates. Okay? If you see that you were having a respiratory acidosis, for example, and then suddenly the circle goes to the other side, this means that you're having another disturbance. Okay? Alright. Now let's take metabolic acidosis, but now I want you guys to help me. Oh, sorry. Someone's asking, why is it called alkalosis if we don't go above the normal pH range when fixing the problem? We call it alkalosis if we... Okay. Do you mean that when we said that the type of compensation is metabolic alkalosis? Is that what you meant? Okay. So when I said what type of compensation is this, I wanted to stimulate your thinking as to what is happening in the body. What is the body trying to do? Okay. However, this doesn't mean that I'm going into a state of metabolic alkalosis. Okay. I'm just saying that the body is doing what would happen in metabolic alkalosis in order to make the pH go back to normal. So your pH is really acidic. Your body's trying to make it alkalotic by increasing your bicarbonate. And so you're going to metabolic alkalosis, but this is not what actually happens. Okay? It's like you're having a problem, so you're trying to balance it by doing another problem. Did you understand my point? Yes. So the pH, okay, at first it was a 7.2, but then that never goes above and beyond 7.4, okay? It's gonna go to 7.35, okay, 7.36. So it's at least trying to reach the normal range, but you'd never go from 7.2 to 7.6 in a compensation. That does not happen. Okay? All right, you're welcome. Okay, now let's try metabolic acidosis. Okay? I want you guys to tell me, what is the primary disturbance going to be in this case? You're having an increase in H plus. Excellent. Okay? So metabolic means it's coming from the metabolic side. Okay? So from the ions. And you're having acidosis, so this means you're having an increase in your H plus. Okay? And what happens when I have an increase in H plus? We said this before. The H plus would bind to bicarbonates and to B minus, and so these two values would decrease, and you'd get more CO2, but CO2 would be exhaled by the lungs. Okay? So, in other words, your buffer bases would go down because you're taking up all the bases, and your pH would go down as well because you're having increased hydrogen. Okay? So this was shown here. All right? This is basically everything that we said. All right, so H plus goes up, pH goes down. What happens in the body is that the bicarbonates will go down, the B minus would also go down because you're binding the H plus. The buffer bases decrease less than normal, and your base excess would be negative. And what does it mean, base excess of negative? It means that the difference in buffer bases from the normal value, the buffer bases are lower than the normal. So this means you're going into acidosis. Okay? What do you guys think is the compensation in this case? What can we do? So we said the problem is coming from what? The acidosis from the hydrogen. So what am I going to do? Yes. So basically, you will try to go into respiratory alkalosis. In other words, what are you trying to do to the carbon dioxide? Do you want to increase it or decrease it? You want to decrease your carbon dioxide. Excellent. Because if you decrease your carbon dioxide, we know carbon dioxide is acidic. So if you decrease it, you're going to try to make the blood more basic. Okay? Okay. So now let's go talk about the Henderson-Hasselbalch equation for metabolic acidosis. So your initial disturbance is that your pH is going down, right? What happens in the body is that the bicarbonate is going to bind all this H plus, and so the bicarbonate goes down. However, does carbon dioxide change? No, because we said it's always exhaled through the lungs, so it doesn't increase. Okay, this is before any compensation happens. All right? So, again, if you have a smaller number in the numerator, your log value would go down, and so this is why the pH goes down. And so you need a compensation again. Now, reminder, again, you cannot compensate a metabolic problem by changing the HCO3 minus, which is the bicarbonate, because this is part of the disturbance. So you can change the carbon dioxide. Okay? Now, this is what happens. The body would signal the lung to do what? If I want to decrease my carbon dioxide, I'm going to start breathing faster. Okay, I'm going to breathe faster, and then this way I'm going to exhale more and more carbon dioxide. So you see the green arrow here, my carbon dioxide goes down. Now, when my carbon dioxide goes down, what happens to my pH? It goes up. Okay, see the ratio here? Now you're having two small numbers over each other, you get a bigger number, you're going to increase pH. Okay? Now, what type of conversation is this? You guys answered it before I even asked you, respiratory alkalosis. However, when I ask you what is happening to this patient, okay, you don't say that they are having respiratory alkalosis. Okay, if they're having a compensation, so you're having metabolic acidosis, right? And you're having a compensation by decreasing your carbon dioxide. Does this mean the patient is having respiratory alkalosis right now? No. Okay, patient's still having metabolic acidosis. It's just being compensated. All right, just please don't mix this up. Okay. Okay. Someone's asking, do they ask it like this by giving us the compensation and asking us what the patient has? They could ask you both ways. So they could give you the problem and ask you what is the patient having. They might not tell you that the patient's compensating. They might just tell you, okay, you're having this pH, this CO2, and this bicarbonate. What's the problem? Okay, they could give you choices of acidosis, metabolic acidosis, partially compensated respiratory alkalosis. Okay, so this is why we're going through each case so that you know what changes in everything, and this is why you need to know what is a primary disturbance. If you know what the initial problem was, this is your disturbance. Anything that happens after that is the compensation. Okay, and we're gonna go through a lot of examples of that. Okay, so now we're going to extrapolate the metabolic acidosis onto the Davenport diagram. Okay, so we said that your change is not in the CO2. Your CO2 is the same. So you're moving along this curved isobar of 40. You're going from this dot to this dot. Now what happens here? Your pH went down. It went from 7.4 to 7.2. Now what happens to your bicarbonate? It also went down. First you were at 24 and now you are at about 15. Okay? So you need a compensation for this. Now what do you guys think would happen to this arrow? Like where can the arrow point in order to compensate it? Okay? Or what do you want to do? You're gonna go right. Excellent. You're gonna go in this direction in order to try to decrease the carbon dioxide. So what am I doing? I'm going from isobar 40 to isobar of 20. So I'm decreasing the the carbon dioxide. Okay, and this would make my pH go back up and make it a bit more closer to normal. And so your respiratory compensation is this. Okay? And that's basically what I'm saying. Now, we're not reaching the normal line yet. So in any case where you're having compensation and you're not reaching the normal line, you call this a partial compensation. Okay? So in this case, this green dot, what do we say about it? Partially compensated metabolic acidosis. Okay? Okay, is acidosis clear? We're done talking about acidosis, okay? Is it clear for everyone? Okay, excellent. All right. Okay. Now, this is about metabolic and respiratory alkalosis. So it's the opposite, but it's technically the same thing. Okay? So I'll go through it briefly. Now, for this first arrow number 1, you have the same isobar of 40. So this means whenever you move along an isobar, this means that you're having a metabolic disturbance. If you're moving with, along the buffer line, you're having a respiratory disturbance. So here you can see that the carbon dioxide is not changing. So your problem is coming from an increase in what? CO2. It goes here, and then in order to compensate that, you want to do what? You want to increase your carbon dioxide. And so you go from 40 to 60, and what happens to your pH? At first it was increased, and now it starts going back to normal. This is for metabolic alkalosis. Now for respiratory alkalosis, we said we'd move along the buffer line. Your CO2 went from 40 to 10, okay? So this means alkalosis and it's respiratory. Now to compensate that, you want to move along the same the same isobar, but what are you going to do? You're going to decrease your bicarbonate. When you decrease the bicarbonate, this means you want the blood to become more acidic, your pH is going to start to go down. Okay? Now I wrote all of this here so that you guys can go through it on your own, but it's basically the same thing as acidosis, just take it the opposite way. Now, these are some points that you want to remember while you're looking at, basically when they give you values in the exam, they're going to give you three values. They're going to give you pH, they're going to give you CO2, and they're going to give you bicarbonate. Okay? Now, you want to also remember that other than these values, they might give you a Davenport diagram like this diagram. Sorry. They might give you something like this. And they'll ask you which dot of base means partially compensated respiratory alkalosis, for example. Okay, so you want to know how you can change or compensate or cause the disturbance in the diagram. And we'll talk about how to do that now. Now, in order to know what kind of change is happening and which one's the disturbance and which one's the compensation, okay, you need to remember these three things. First, remember that a respiratory disturbance happens along the buffer line, the red line. I've said that before. If you're having the circle on the red line, this means that it is a respiratory disturbance without compensation. Now, second is for the metabolic disturbance. Metabolic disturbance happens along the same isobar, okay? It means that my CO2 is not changing. What's changing is my bicarbonate, okay? Next is the idea of overcompensation. Again, you can never overcompensate, okay? So the compensation never crosses the dotted line, which means the pH of 7.5. Now we're going to take multiple examples of that, but again, this is the red buffer line, okay? So what do you see here? What's changing? The carbon dioxide. So what type of change am I having here? What is it? Is it respiratory or metabolic? What do you guys think? It is respiratory. Excellent. Okay, so you're having a respiratory disturbance without compensation because you're still on the buffer line. Okay? Now, if your pH, now how do you know if it's acidosis or alkalosis? You check this line and you see which side you're on. If you're on the left side with a low pH, it's acidosis. On the right side with high pH, it's alkalosis. Okay, same thing over here. Okay? You're having the change along the same isobar of 40. Okay? So what kind of change is this? Is this respiratory or metabolic? Excellent. It's metabolic. Why? Because my CO2 is not changing. What's changing is the bicarbonate only. So this is a metabolic disturbance without compensation. Again, pH less than 7.4, so these 2 points would be metabolic acidosis, these would be metabolic alkalosis. Okay, now we're going to practice. I think this part is going to be a bit fun. Okay, now let's do that. Here you have this red dot. Literally, this is what the exam is going to tell you. It's going to give you this picture and it's going to tell you what is the problem. Okay? So, what do you guys think is the issue over here? Okay? Or how can we try to reach this problem? Is it respiratory or is it metabolic? Can you take a wild guess? Now we can tell that it is acidosis, right? Because we're on the right side, or on the left side. Okay, you guys are saying respiratory, which is excellent. Okay? And it's compensated. Correct. Okay? Partially compensated respiratory acidosis. Excellent. Okay? Now let's see how we got that. Now, we can think of these lines, so the buffer line, the green line, as roads. Okay? And think of the dotted line as a block in the road. Okay? So there are two ways you can reach this red line from the normal. You can either go that way, okay? So you go along the buffer line, so meaning you're inducing a respiratory acidosis, and then you try to compensate that. Or you can go the opposite way. You can go along the green line, the isobars, so you're doing a metabolic problem, and then you're going back to the red dot. Now, do you think which of these ways works? Okay? Or do you think, in other words, does the red arrows work? No, you cannot cross the dotted line. So this pathway does not work. So the only one that works is the other way. So you are having a partially compensated respiratory acidosis. Excellent. Okay, what about this red dot here? This other one. Can you guys tell me what kind of disturbance are you having, and are you having a compensation or not? We know it's acidosis. It's on the lower side of the pH. But is it respiratory or metabolic? Now this one's a bit tricky. Okay, you're saying it's both. That's correct. Okay, now how does that work? We have two pathways again. We can either move along the buffer line, and so we have respiratory acidosis and then compensate that, or you can move along the isobar and go to the red line. Now, are you crossing the dotted line at any of these points? No. So this is weird because you can go both ways. So what does this mean? You have both types of disturbances, and you're having, sorry, this is what we talked about, the partially compensated respiratory acidosis in this slide. And this is what we mean by combined respiratory and metabolic acidosis. You're having both disturbances, and yes, you can have that, okay? Someone's asking the paths in the graph, it's showing either partially compensated respiratory acidosis or a worsening metabolic acidosis, which is impossible. So we say that it's the former, yes. Okay? You're talking about this section. Okay? How can you tell if it's both? If it's not crossing the dotted line. Okay? So in this top case over here, exactly, like you said, if the lines doesn't come, they don't cross. Okay? So basically, in this top part where we had respiratory acidosis, okay, you went this pathway, the blue pathway, and it works. But when you try to go to the red pathway, first of all, you're basically doing an alkalosis, and then you're trying to go back to acidosis. That doesn't work. And you're crossing this line. So you don't want to cross this line, okay? So this is why you only chose one of the pathways. However, in the second condition, you went respiratory acidosis, and then you went down to compensate it. But you also went as what? Metabolic acidosis, and you tried to compensate it. And both pathways are not blocked by the normal pH line. They're not blocked by the dotted line. So this means that they both work. Okay? And there's also another tip that I'll give you later on how to know quickly if you're dealing with combined or just one type. All right? So again, this is me explaining what happens in those slides. All right. Now, this is basically, you're going to see this in the exam. They're basically going to they're going to tell you which dots of these means this. Okay? So, let's try to take one and do it together. All right? Let's take this one. This dot over here. Can you guys tell me what type of disturbance is it? So, a tip to start off is think of it if it's acidosis or alkalosis. Now, it's clear that this line is alkalosis because its pH is high. Now, is it respiratory or is it metabolic? It is both. Excellent. Okay. Now, another quick tip is that if it's along the buffer line, if the dot is on the buffer line, this means that it is not being compensated. It is a respiratory change without compensation. If it's along the isobar, okay, this normal green line, then it's also not being compensated. However, if it's anywhere else, then that means there's a compensation. So in this case, yes, you're right, both work, okay? You can either go respiratory, alkalosis, and compensate, or metabolic, alkalosis, and compensate. And you're not crossing the dotted line, so it's okay. Okay? And again, you can take this figure, you can practice on your own, all right? Yeah. Now, these are some tips that I'll give you guys, okay? So you can use the three lines to determine what type of disturbance and compensation you have. For the dotted line, this means a normal pH. The buffer line, anything along this means respiratory disturbance. Anything along the green line, which is isobar 40, is a metabolic disturbance. Now, if the circle, so whatever thing you're having, if it's to the left of the dotted line, so on this half, it's acidosis. If it's on that half, it is alkalosis. Okay, some other tips are, we want to look where exactly this circle is, or if it's between what lines, okay? So we talked about these three important lines. Now let's say the circle is between this dotted line and the buffer line, okay? You can immediately conclude that this is a respiratory problem with a partial compensation, okay? And if you try to take any dots and you try to do the arrows thing, you'll see that that's what happens. Now let's say the dotted line is between, the circle, sorry, is between the dotted line and the green line. What kind of disturbance are you going to have? Metabolic with partial compensation. Okay, so it would be somewhere here or somewhere here. Now, I'll ask you guys, what about if you had it between both the buffer line, so the red line, and the green line? So something like this here or this here. What kind of disturbance are you having in that case? You're having a combined respiratory and metabolic disturbance. Correct. Okay? This is a picture that basically shows you how you can differentiate these areas. So these are the areas that we said. Between the dotted line and the red line, you're having what? Either respiratory acidosis or respiratory alkalosis. Between the dotted line and the green line, you're having a metabolic thing. So it's either metabolic alkalosis or metabolic acidosis. Between the green and the red, you're having combined, either acidosis or alkalosis. Okay, everything clear? Okay, excellent. You're welcome. Okay, now, do you think a patient is going to come into the clinic with a Davenport diagram like this thing? They don't do that, okay? And that is not what happens in real life, okay? So, what's going to happen is that you will be given lab values, okay? So, and you're going to know from these three lab values what's happening with the patient. Excuse me? Okay. So the three parameters that are basically given to you by the lab, these are the pH, the PCO2, and the bicarbonate or the base excess. So for pH, let's stick to the number 7.4 because this is what we'll mainly be using in our exercises. So 7.4. Less than that is acidosis, more than that is alkalosis. Okay? Alright. Now for PCO2, we said the normal value is about 40. Anything less than that is respiratory alkalosis. Anything more than that is respiratory acidosis. For the third parameter, you either have bicarbonate or the base excess. Now for base excess, the normal base excess is 0. I mentioned that before. However, if your base excess goes up, that means you're having more base, and so you're having a metabolic alkalosis. If it goes down, this means metabolic acidosis. Now let's say we're using bicarbonates. What is the value on which is normal, basically, for bicarbonate? I want to see if you guys remember. What's the number that we use for bicarbonate? 24. Excellent. Okay. So let's say you don't have a base excess, but you're given bicarbonate, then you would see if it's 24. If it's less than 24, then you're having what? Acidosis or alkalosis? If it's less than 24. Okay. You're having acidosis. Excellent. And if it's more than 24, you're having alkalosis. So keep these three parameters in mind, and this is how most of the questions come. Okay. All right. Now we're gonna go to court. Okay. We're having pH being the plaintiff, and it's saying that okay, I'm having this issue, which is either acidosis or alkalosis, and now I'm accusing him of doing this. Now pH is gonna accuse two people. It's gonna accuse the CO2, so respiratory, or it's gonna accuse the base excess or bicarbonate, which is the metabolic. Okay? And those are the defendants. So we want to know who's saying the truth and who's not. Now keep in mind that if both parties are saying the truth, then this is the disturbance. However, if both parties have different answers, so the pH is saying I'm in acidosis and the PCO2 is saying I'm in alkalosis, this means that the defendant is trying to cover up the problem, it's trying to cover up what's happening, and this is decompensation. Okay? So always see who's lying, the person that's lying, that's decompensation. If it's saying the truth, then this is the disturbance. Oh, now I'm going to take some cases and I want you guys to answer. Okay? You're having a pH of 7.2, your PCO2 is 60, and your base excess is 0. Let's start off. We want to start off systematically. Don't directly tell me that this is metabolic acidosis or alkalosis, compensated or not. Let's take it step by step. What do you think of the pH? Is it acidosis or alkalosis? The pH is acidosis. Excellent. What about the PCO2? Is it telling you acidosis or alkalosis? So basically respiratory acidosis or respiratory alkalosis? It is saying respiratory acidosis. So it is high, it's saying the truth. What about the base excess? It's 0. What does that mean? Base excess is normal. Excellent. Okay? So you're not having compensation. And what are you having? Uncompensated respiratory acidosis. Okay? Again, we saw that the PCO2 was telling the truth, so this was the disturbance. Okay, case number 2. Same pH, same PCO2, but now the base excess is positive 10. Okay? We said this is acidosis, and we said that the PCO2 is telling the truth, so this is respiratory acidosis. But what about the base excess? It's positive 10, so it's much higher than normal. So what does this mean? The base excess is lying. It's saying that it is in metabolic alkalosis. So this is the compensation. So how are we going to say it? Partially compensated respiratory acidosis. Now, why didn't we say fully compensated? Can anyone guess why? We're always saying partially. Why are we not saying fully compensated? It's never fully right. Exactly. The pH never corrects. Okay, exactly. If it was fully compensated, the pH would go back to normal. It would go back to 7.2. Okay, excellent. All right, so case 3. pH is 7.2 again, PCO2 is 60 again, and your base excess is negative 10. So acidosis, CO2 is telling the truth, but what's happening with your base excess? It's low. Okay, your base excess is also telling the truth, and the same with metabolic acidosis. So you're having both saying the same thing. Exactly. They're both being truthful, and so it is a combined respiratory and metabolic acidosis. Excellent. Okay, case 4. We're having a pH of 7.2, but now the PCO2 is low. It's 30, and your base excess is high. It is 10. Now the pH is acidotic. What about the PCO2? What is it telling me? Is it respiratory acidosis or alkalosis? It is respiratory alkalosis, so it's not telling the truth. Keep that in mind. What about the base excess? What is it telling me? Base excess is high. So it's also saying alkalosis. What's going on? They're both not saying the truth, right? What do you guys think is happening in this case? And don't worry, there's nothing wrong with you or your analysis. But what is that? This is what you got back from the lab. These are false values. Excellent. There's something wrong with the lab itself. The lab results are false. Okay? And so you need to send the sample again back to the lab. Okay? Right. Case 5. pH is 7.2, PCO2 is 30, and your base excess is negative 10. Now you guys already said this would be acidosis. The CO2 is showing what? Respiratory alkalosis. And the base excess is showing you metabolic acidosis. So what are we having here? Okay, someone's saying if pH is what decides acidosis or alkalosis, PCO2 is agreeing it's respiratory, base excess is agreeing it's metabolic, and if both PCO2 and base excess agree, then it's combined. If one of them disagrees, that means it's partially compensated. If both disagree, the results are excellent. Okay, this is basically an excellent summary of what we've been doing in the exercise. Okay, case 5 is partially compensated metabolic acidosis. Excellent. Okay, I'm very proud of you guys. You guys are on top of the game. Okay, now let's dig into some special concepts. I know we've taken a lot of time, but we're almost done. Okay, now there's a dilemma when it comes to the bicarbonates increase in respiratory acidosis. Why is that? Now we know that the primary mechanism is that the pH goes down because your CO2 goes up. And we also said that bicarbonate does go up a little bit, right? However, we don't know if this increase in bicarbonate is happening because of the disturbance itself or if it's the compensation. Okay? So the red arrow shows just from the disturbance itself. The green arrow shows that this is a compensation. So how are we supposed to know which one it is? We can know this by knowing the normal increase in CO2 in bicarbonate from the disturbance. Okay? So in other words, I want to look at this Davenport diagram, right? If you go from point 1 to point 2, you see that the bicarbonate at first was 24, and now it is 30, okay? Now, does this increase, what do you think? Is this the compensation, this small increase, or is this coming from the disturbance itself? What do you guys think? It is coming from the disturbance itself. Correct. Okay. Another tip is that the circle is still on the buffer line, so there's no compensation. Okay. Now this basically change from 24 to 30 is a 6 millimole increase. Okay. And now this 6 millimole has changed over a pH of what? Over 0.2 units. So at first you have 7.4, but then over here you have 7.2. So 0.2 units of pH basically changed the bicarbonates by how much? 6. So how much bicarbonate would change if you're only having a 0.1 change in pH? What do you guys think? So you said for 0.2, you're having a 6 millimole increase in pH in bicarbonate. What about a 0.1 unit? Excellent. It would just increase by 3, okay? So in other words, if the increase in bicarbonates is more than 3, okay, in the setting of respiratory acidosis for 0.1 pH, then you know that this is a compensation. So for example, if you have 7 instead of 3, then you know that this is a compensation, okay? Now, the reason why we prefer using base excess is that because we don't face this problem, okay, with base excess. Base excess, it's either more than normal, less than normal, it's clear, okay? But with bicarbonates, you're not sure, oh, is this the actual, is this coming from the disturbance itself, or is it the actual compensation? Okay, last concept, okay, which is the anion gap. Now, keep in mind, the anion gap is only used in metabolic acidosis. Please do not be confused. Okay? All right, someone is asking, we'll go back to the previous slide. If it's greater than 3, it's compensated. Okay, keep in mind, if the bicarbonates, I'm saying this is over a pH of 0.1 units. Okay, so if I go from 7.4 to 7.3, I expect the bicarbonate to increase by 3 only if it's not compensated. However, if I went from 7.4 to 7.3, and for example, my bicarbonates increased by like 10, this means that I'm having compensation, okay? But that doesn't mean that 3 is the fixed value, okay? So if you're having your pH changing like 0.4, then you need to multiply 3 by 4. So if you change the bicarbonates by up to 12 units, that still doesn't mean a compensation. It's just because of the disturbance, okay? But this was just applying to the pH of 0.1. Okay. So again, you're welcome. So again, anion gap, we said this is only used in metabolic acidosis. Please do not get confused. Okay. Just remember, anion gap is just metabolic acidosis. It is not for any other disturbance. Okay? Okay. So what is an ion gap? Basically, if you look at the electrolytes that you have in your plasma, so sodium, potassium, chloride, bicarbonate, magnesium, calcium, the positive ions, so the cations, are equal in number to the negative anions. They always have to be equal, okay? Or that means your blood would be charged and you start electrocuting everything, which is crazy, okay? So there is no anion gap in real life, but we use it to interpret lab results, okay? Because why? There are some values that we cannot measure. Okay, some cations and some anions that we cannot measure. So we do have this gap that we see in the lab results. Now for cations, what are the cations that we measure? We mainly measure sodium and potassium. Other cations, like magnesium and calcium, their concentrations are very, very low, so they don't really affect the values. For anions, we measure chloride and bicarbonates. However, a large portion of anions comes from proteins like albumin and other things, but these are not measurable when we check for an anion gap. So this is why usually when you have an anion gap, your cations show that they're more than the anions. Okay? But this doesn't mean that the blood itself is having this deficiency, it's just that you can't measure those values. Okay. Now, how do we calculate the anion gap? We can either calculate it with potassium or without potassium. Let's say we calculate it with potassium. So this is the formula. You take the positive ions, you add them, and you subtract that by the sum of the negative ions. So basically sodium plus potassium minus the sum of the negative ions, which are chloride and bicarbonate. Okay? And you can see here in this figure that you have sodium. Sodium makes up most of the cations, the positive ions, and it's about 144 normally. For the negative ions, you have chloride is making up most of it, and the big portion is also coming from bicarbonate. And then these are the ones that we cannot measure. The proteins like albumin, sulfate, organic acids, you cannot measure these in the lab regularly. And so this forms the anion gap. Okay? Now, normally, when you test with potassium, the anion gap is 14 to 18. Okay? Now, why am I saying with potassium and I'm specifying this? Because if you're not using potassium, let's say you didn't test for potassium, you just tested for sodium, chloride, and bicarbonate. Okay, so you removed potassium from the equation. Now you have to account for that change when you talk about the range of normal anion gap. Now, potassium itself is about 4 like millimoles, okay? So if I remove potassium from the equation, I have to subtract my range by 4. So at first over here with potassium, I said it is 14 to 18. Now what am I going to do? I'm going to say that without potassium, normal anion gap is 10 to 14. Okay? Did you guys understand this concept of an anion gap? Yes. Okay. Excellent. Okay, why did we subtract 4? Is it fixed? Okay. So potassium, the normal value of potassium is about 4. Okay, so here you see sodium is 140, chloride is 105, bicarbonate is 24, 25. Okay, so potassium is about 4. The actual range is 3.5 to 5, but we take the median value, which is 4. Okay? So if I'm calculating the anion gap with potassium, okay, I said the normal anion gap is going to be 14 to 18. But let's say I removed potassium, so I'm technically subtracting this anion gap by 4. صح? لأنه I'm removing the value of potassium. So if I remove that, I have to make my normal anion gap basically account for the absent potassium, and I removed potassium from the equation. Okay? And so that's why I kind of subtracted the range by 4. So it says 14 to 18. Now, without potassium, we would say it is 10 to 14. Okay? Did you understand that concept? Okay, excellent. All right. When we talk about anion gap, we have what we call high anion gap and normal anion gap. Okay, so metabolic acidosis can either be high anion gap metabolic acidosis or HAGMA, and normal anion gap metabolic acidosis or NAGMA. Okay, what is the difference? When it's high anion gap metabolic acidosis, this indicates that your problem is coming from metabolism itself. Okay, now if you look at this picture over here, this is the normal anion gap. Okay, without metabolic acidosis, you're having sodium, you're having bicarbonate, chloride, and this is your anion gap. Now over here that you see, your anion gap increased, right? You see the red space is more. Your chloride is the same as over here. However, what decreased your bicarbonate? And remember we said in metabolic acidosis, sometimes your bicarbonate decreases, okay? And so you're having a high anion gap, so an increased anion gap, metabolic acidosis. Okay, now these are some causes. This is a mnemonic that you could use later on when you're in third year for the causes of high anion gap metabolic acidosis called MUD. You don't need to memorize this story. No one's going to ask you what causes high anion gap metabolic acidosis. What's the difference between that and normal anion gap metabolic acidosis? Normal anion gap metabolic acidosis means that your problem is coming from the GI tract. It's not coming from metabolism or from the kidneys. So basically, you're losing negative ions from the GI tract. Now, why is the anion gap normal? We said that you do lose bicarbonate in metabolic acidosis, right? However, if you look here, what happened to the chloride? It's increased, okay? Now, why did the chloride increase? This basically happens in the gut to compensate for the loss of bicarbonate. So if you're having diarrhea and you're losing bicarbonate in the gut, what the intestines try to do is that they try to retain chloride, okay? And so when you retain chloride, you basically balance the loss of bicarbonate. And so your anion gap stays normal, okay? So in vomiting, the concept is the opposite. In vomiting, you're losing a lot of chloride, okay? And so you get metabolic alkalosis. I'm not going to go into details of that. But in vomiting, you also lose a lot of acid because you're vomiting, it comes from the stomach. The stomach has acid, it has hydrogen. And so this is just an interesting fact. So when you vomit, if you vomit a lot, you could go into metabolic acidosis. Sorry, alkalosis because you're losing acid. Okay? But I just want you to understand the difference between the high anion gap and normal anion gap, what happens to the bicarbonate and the chloride. Now this is, again, another mnemonic as to what causes normal anion gap metabolic acidosis. However, again, you do not need to memorize it. It's just for your information. Okay. Now, we've talked about how to identify metabolic acidosis and everything about it. Now, how can we correct it? Okay. You want to give these patients bicarbonates to raise the blood pH, which makes sense. They're losing, what are they losing? Sorry, they're gaining hydrogen. So you want to give them bicarbonates in order to bind that hydrogen. Now, keep in mind that you don't want to overcompensate patients. Now, we said the body never overcompensates, but let's say you give them bicarbonate, and you keep on giving them bicarbonate. They couldn't go into alkalosis. Okay, so that's when we need to calculate the amount of bicarbonate that we give. Now, we usually give them you don't give them bicarbonate as an ion, you give them sodium bicarbonate, okay, as like IV fluids. But this is the equation that we use. Okay, so the base required, so the amount of bicarbonate that you would give them is equal to this formula. You'd have the negative base excess, okay, so remember that value, times 0.3, which is a constant, times the body weight in kilograms. So let's take this example. This patient has a base excess of -10, and we said the constant is 0.3, and the body weight is 70 in kilograms. So if you multiply 10 by 0.3 by 70, how much do you get? 210. And this is the amount of base that you need to give the patient. And so you give the patient 210 milliliters of sodium bicarbonate. Okay? Did this one make sense? Okay, excellent. All right. Someone's saying, is it negative because it's acidosis? Yes. Your base excess is negative because you're in acidosis, correct? Okay. Now we talked about how to correct metabolic acidosis. Now how do we correct metabolic alkalosis? It would definitely be, again, using a formula. And this time you want to give them something acidic, okay? So you calculate the positive base excess times 0.2 times the body weight in kilograms, okay? Now, uh, what the things that you could give them are arginine with chloride or lysine with chloride, okay? You don't really give them hydrochloric acid because um, that would be, that has a lot of side effects. It's pretty dangerous to do that, okay? So you usually give them these two molecules. Now, do you have to memorize this? I don't think they really ask this. It's pretty low yield, but I think you should at least just know the formulas of how to correct either metabolic acidosis or alkalosis. Okay? Okay. I just have three practice questions if you guys can bear with me. Straightforward multiple choice questions. Okay? Now, this is the first question. Basically, you have a patient that was admitted in a coma, and they gave you the lab values. You have a PCO2 of 16, bicarbonate of 5, and a pH of 7.1. What is the acid-base disorder that you're having right now? Okay, someone said A. Okay, yes, you guys are correct. It is A. Okay, it's metabolic acidosis. Why? First thing you look at is what's pH. pH is low, so there's acidosis. So immediately cross out options B and D, you have A and C. It's either metabolic or respiratory. Now you look at what the CO2. The CO2 is low. So it's saying that it's in respiratory alkalosis. So it's not telling the truth. So this is not the disturbance. This is the compensation. You see the bicarbonate is low, and this is metabolic acidosis. Next question. A student is nervous for a big exam. She's having, let's say, a panic attack and she's hyperventilating. What do you expect to happen? Like which which acid-base disorder do you expect her to have? Okay, I'm getting D as an answer. Respiratory alkalosis. Excellent. That is D. Okay, why is that? Because she's breathing a lot, so she's exhaling too much carbon dioxide, and so you're going into a respiratory issue, and you're getting rid of CO2, so that makes the blood more basic. You get respiratory alkalosis. Last question. Okay. Yes, exactly. Hyperventilation is what causes the increased exhalation of CO2. Okay. Now this last question. All are true for renal handling of acids and metabolic acidosis, except which of the following? Now this goes back to that figure of the tubules and when we had the excretion and reabsorption. So people are saying B. The bicarbonate's reabsorption is decreased in metabolic acidosis. You're correct. Okay? All right. Okay. Why is it B? All are true for renal handling of acids and metabolic acidosis, except. Okay. So basically, I'm asking you which of the following is false. Okay? I'm not asking you which one is true. Okay? So see, look at which ones are actually happening in metabolic acidosis. Does the hydrogen ion secretion increase in the urine when we have metabolic acidosis? Yes, because you're trying to get rid of hydrogen. For C, because you're excreting a lot of hydrogen in the urine, does the acidity of urine increase? Yes. Okay. For ammonia, does it increase in the urine? Yes, because remember NH4 plus goes out into the urine. Now for B, it's saying that the bicarbonate reabsorption into the blood is decreased. Okay, is that true? No, because we do, relax, you do increase the reabsorption of bicarbonate, okay, so that you can make the blood more acidic, more basic. Okay, you got it? You're welcome. All right, we are done, and thank you so much. I know this took much more than scheduled. It was supposed to be an hour and a half, but I really hope that you guys, you know, were able to understand. You're welcome. And this is a QR code. You can scan it, and it has a feedback form. Okay. As for the PowerPoint, they will send it to you guys. Okay. So you'll get the PowerPoint sent to you. Okay. All right. Okay. So we're done. Have a nice day. And yeah.